So, thank you very much, and thank you for the invitation to Greece. It's a real pleasure being here and meeting old friends again. And I was never really his chief. He was independent already, and he's his own chief now. So, the lecture for the next half hour will be on why kidneys fail and how much can we learn about this in terms of new therapies. So, I think today we have come to realize that the most important step in why kidneys can fail, and in particular glomeruli fail, is this central step here, podocyte damage, where if you go to a textbook today, it will tell you the podocyte is a terminally differentiated cell, and if you lose it, you cannot replace it, full stop. Well, this may be true, and in fact, we have good evidence that this is a key cell, but there's a new cell coming to attention, which is the parietal cell. And I will tell you a little bit about this cell, about which in the past we have essentially known nothing. So the first evidence that podocyte damage is really a bad thing, the first piece of evidence, there's a lot of evidence, is this paper where Don Shokayashki's group from Vienna, and we have shown that if you induce a particular microRNA, it doesn't matter why this one and what it does, but if you overexpress it in podocytes, that then you have this massive phenotype, and it only takes 10 weeks and the whole glomerulus is gone. We had actually known this for a long time. This is an old study of ours, which is 16 or 17 years old, where we had done a very simple experiment. We had used a model of membranous nephropathy. You see that normally these red glomeruli look fairly normal by histology. And we had given them a growth factor, fibroblast growth factor 2. And again, I will save you the rationale why we did this. But then something unusual happened. Podocytes went into mitosis. Now your textbooks say podocytes cannot do this. If you ask your pathologists, they will tell you that even in a human biopsy, occasionally they see a podocyte in mitosis. But think about this. What does a cell do when it goes into mitosis? First, you divide the nucleus, but then comes the problem. Then the cytoplasm has to divide. In order for the cytoplasm to divide, a cell has to detach from the matrix. This is no problem in the skin. There's lots of matrix around, but it's a huge problem for a poor podocyte. So if you think about this, what a podocyte can do is, if they go into complete cell division, this cannot happen because the podocyte will simply fall off the glomerular basement membrane and it will be excreted in the urine. So what will really happen is that this podocyte will divide, it loses adhesion, and that is a stimulus for apoptosis or podocyturia. Or it only does half of the job and you have nuclear division and you end up with binuclear podocytes, which is observed in some diseases like FSGS. Indeed, if you look hard enough, you can find podocytes in the urine and you can culture them and they live. And the interesting thing here is when we induce this model, aminonucleoside nephrosis, a toxic injury to the podocyte, before the proteinuria starts, see proteinuria starts around here between days 3 and 5. Before you can detect that, podocytes are excreted in the urine, and please note that this is a log scale, so this is a massive increase, and it increases 1,000-fold in the massive disease. But the important thing and observation here was that podocyturia quickly decreased again and came down, whereas proteinuria persisted. So the hope associated with this finding is that we can use podocyturia to detect ongoing glomerular damage and to guide the clinical treatment to the phase of damage, as opposed to proteinuria, which could be active damage or simply a scar. In 5/6 nephrectomy, the situation is different. Proteinuria increases, and of course you have continuous damage to podocytes, and it becomes really severe late in the disease. Now the scary thing is that even in you and me, this morning when we went to the toilet, we have excreted podocytes, all of us. And if you quantify that, the number of podocytes you excrete this morning every day, you can calculate that by the age of 45, we have no podocytes left. We should all be, well, some young people not, but I should be on dialysis if I don't have a mechanism to replace my podocytes. So the textbook, simply from this observation, must be wrong. Podocytes must have a way to recover, to divide, and to repopulate, otherwise we would not live beyond the age of 40 without dialysis. And the big excitement was when we discovered this. This is, just to illustrate what we have done here, is we have genetically labeled parietal cells. We came across a promoter that is only active in the parietal cells. So we were able to genetically label these cells and make them blue forever. So you can now trace these cells. And when we took these young mice and we let them live, all of a sudden we discovered blue cells on the glomerular tuft. And we said, wonderful, we have found the stem cell of the podocyte. And podocytes are continuously repopulated by the parietal cell. And indeed, this was quite marked. If you look at this, the number of blue cells per 100 glomeruli, it was around 2 cells per glomerulus in this young mice, if you simply follow them into adult life. An Italian group made a similar observation, and we both thought we have found it. We were a little more cautious. We said there's recruitment, whereas Paola Romagnani group already called this regeneration. Now there's a problem with this observation, and it simply is not the truth. There's an alternative hypothesis that as glomeruli develop, there's a committed cell population, the green cell population, which is committed to become podocytes and which simply migrates onto the glomerular tuft as the tuft expands and folds, and then you have this razor-sharp separation between parietal cells and podocytes. If you look under the electron microscope, it's fascinating. It is one cell is a parietal cell, and the next one is a podocyte in everybody of us. And of course, this would then imply that at some point in adult life, this simply stops because all committed cells have migrated onto the tuft. And this is indeed what happens. We did the same experiment in adult mice. We labeled all the parietal cells, and you can see that even the last parietal cell here is labeled. But even if you wait for 18 months after that, you will not find parietal cells on the glomerular tuft. So there's a major difference between young adolescent animals, where you still have glomerulogenesis and postnatal glomerular development going on, and adult people. And the take-home message from this study is that in all likelihood parietal cells are not stem cells and not the progenitors. We then asked what would happen if I stressed the cells. We simply took out one kidney, did a uninephrectomy, or did a 5/6 nephrectomy, and you can see that the percent of positive cells doesn't change at all. Nothing happened. So, in essence, the question why you and I excreted podocytes this morning and why we are not on dialysis is still open. What we now know is that podocytes indeed disappear when you have glomerular scarring. You can nicely show that if you label your podocytes blue, this is now podocytes labeled, they are displaced from the FSGS lesion. Here's the scar, and they simply disappear. So far, the theory said that podocytes are damaged, they disappear, and this is how you develop a scar. But what we found when we labeled parietal cells, so here blue is now parietal cells again, and you induce FSGS. Here's a focal scar. All for a sudden you see parietal cells on the glomerular tuft, and in the late stage all the podocytes seem to be gone and there's only parietal cells. And here is a particular matrix component that they produce, and it's all parietal cells. So this fundamentally changes our thinking. FSGS does not develop because you damage podocytes. FSGS seems to be an active step where parietal cells are activated and they conquer the glomerular tuft and take over the tuft and displace podocytes. So this is what it would suggest based on a lot of experiments we did, that parietal cells get activated, they form a small bridge onto the glomerular tuft, they displace podocytes, and they take over the podocyte, and this is how FSGS lesions develops. So a primary insult leads to podocyte damage, this crosstalk is disturbed and parietal cells are activated, they migrate onto the tuft, and they deposit matrix and do nonsense. This has a major therapeutic implication. The question then is not so much, how can I prevent podocyte apoptosis? The question now is, how can I prevent activation of these cells, and which mediators are involved in this crosstalk? And there's a lot of very interesting mediators, and I'm certain we will see new therapies in the future based on this. In addition, a really exciting study by Peter Pertidis in the US came out where they showed nanotubules. Podocytes and parietal cells can talk to each other, and they have like the old-fashioned telephone line. So this cell has a very, very small tubule extending to a podocyte, and they can exchange signals. Not only soluble signals, but direct cell-to-cell signals, and this further stresses the importance of why and how the Bowman's capsule talks to the podocyte. This is not a single cell. So I think there's accumulating evidence now that podocyte damage, and in particular parietal cell activation, is a crucial event in the formation of focal segmental glomerulosclerosis. And the question why you and I don't need dialysis is still open. Issue number 2 is many diseases, of course, like IgA nephropathy will start in the mesangium. And whereas in the past mesangial cells were a hot topic, they seem to be the past now. Everything is podocytes or parietal cells. But I think we should not neglect these cells. And one of the most important systems there is the platelet-derived growth factor system, where in particular the ligands B and D and the beta receptor seem to be of crucial importance for the glomerular mesangium. Why can I say so? Because if you delete PDGFB chain, these mice are born, and the young mice are happy for about 1 day, and then they die very quickly. And if you look at the young mice, the one major defect they have is, this is their glomeruli. And what you have there is a large capillary with an endothelium, podocytes, and a GBM, but the complete mesangium is lacking. So at one point in our evolution and in our ontogenesis, PDGF must be absolutely central, and only PDGF-B chain must be absolutely central to develop a mesangium. And of course we know that many diseases and many disease processes recapitulate ontogenesis. So it was not really unexpected that if you antagonize PDGF in mesangial-proliferative nephritis for just a few days, your kidney looks happy again, whereas the control kidney shows all evidence of a failing, scarring kidney. We also could show that the second ligand of that receptor, PDGF-D, if you antagonize it, your kidney looks happy, whereas the control kidney shows all the evidence of a failing kidney. Now, one thing we could not explain in the past, and this is where things come back together again. Podocytes can produce lots of PDGFs. And we could never understand in those days, you see, this is old data, we could never understand how can the podocyte produce PDGF, and how can this act inside the glomerular tuft? Why isn't it flushed away into the urine? And we finally understood it. PDGF made by the podocyte can enter the glomerulus, and the podocyte can tell the mesangial cell what to do. And we understood this by overexpressing PDGF-D in podocytes, very specifically. This is what you get. You get crescents. So this is an evidence of podocyte parietal cell crosstalk. But you also get proliferative lesions inside the glomerulus. And indeed, if you look at PCNA positive cells per glomerulus, they are markedly increased. So a growth factor that is made by the podocyte can act both on the parietal cell, on Bowman's capsule, and inside the glomerulus. And maybe Don Shokayashki in Vienna is right, that the podocyte is the king of the glomerulus. He tells every other cell what to do. And maybe this is why they are so important. And we have many tools now to interfere with that system, including rather non-specific agents like Gleevec. Gleevec, of course, is a non-specific, let's say, dirty tyrosine kinase blocker, which blocks many tyrosine kinases, but there are better ones, more specific for the PDGF receptor that we can use. The only unfortunate thing is that whenever I go to industry and I tell them, look, we have this beautiful target, they have two questions. What is your endpoint and how long does it take? And I tell them the endpoint is loss of kidney function, and if you do a clinical trial, it will take a few years. And the standard answer is, thank you, Professor Floege, it was really nice talking to you, bye-bye. Unfortunately, pharmaceutical industry is not interested in this, and I have learned the hard way that they have beautiful agents against PDGFs, but they don't want to spoil their big business in oncology, where the endpoint is mortality after half a year or so. It's easy to do a clinical trial, and industry often is afraid that they might discover a safety signal in nephrology which could impair the oncology sales. Unfortunately, this is reality. So that was mesangial cells, but there's a third cell, of course, the endothelial cell. What about them? They are kind of neglected because it's so difficult to study them. What happens if you have damage there? Well, again, we did an experiment many years ago where we did something very simple. we antagonized very specifically the key growth factor for endothelial cells, vascular endothelial growth factor, the 165 isoform. And we did this in a situation where the whole glomerulus is destabilized, where you have mesangiolisis in this mesangoproliferative disease model. And we antagonized it for 4 days only, and only just 4 weeks later, half of the glomeruli are scarred, there's massive proteinuria, and you have extensive tubulointerstitial damage. So by simply taking out one crucial endothelial growth factor for just a few days in a vulnerable phase, we kill the kidney. And we thought, well, why not give VEGF? But that's very tricky business. If you inject VEGF, you get leaky endothelial cells all over and you develop severe edema. This is why we got so interested in this one. Studies came out suggesting that another isoform of PDGF, PDGF-C, is a very potent angiogenic factor and maybe much safer than VEGF for clinical purposes. And indeed, this is what we tested. So we antagonized PDGF-C in a model of thrombotic microangiopathy, and we saw that the moment you give the antibody, weight loss is higher, creatinine is higher, injury is higher. Or to turn things around, if you were to give PDGF-C in this model of thrombotic microangiopathy, you will improve your disease, and maybe one day we will treat hemolytic uremic syndrome with angiogenic factors like this. So those were the key cells in the glomerulus. And if you have injury there, obviously the consequence is proteinuria and or a process called misdirected filtration, where you directly filter into the renal interstitium. Again, in the interstitium, this system may be of relevance. Why do I know this? Because this system, the other ligand and the other receptor, seems to be very important in the kidney. I can say that because we know, if you look at a newborn mouse, here's a mature glomerulus, here's the immature glomeruli, here's the nephrogenic blastema. Mice still have kidney development for a few days after birth. If you delete PDGF-A, nothing much happens. You have a nice interstitium. If you delete A and C or the receptor, there is no interstitium. The renal interstitial never forms. And this to us again suggests that this must be something incredibly important. And indeed, we had noted very early on that your and my kidney interstitium is loaded with PDGF alpha receptor. Everything that's brown here is PDGF alpha receptor. If you have a failing kidney, it's a solid mass of PDGF alpha receptor, and you can even trace interstitial fibroblasts invading the glomerulus by their alpha receptor expression. PDGF-C is not there normally. So you have a nice system where the receptor is always there, and PDGF-C, the ligand, only comes in when you have invading, infiltrating leukocytes. This may be one of the key actions, how leukocytes in the damaged interstitium work. And if you antagonize PDGF-C, you markedly reduce kidney fibrosis in this model of unilateral ureter ligation, where the primary insult is to the interstitium. Now, this, of course, is not the only thing that we can think of in terms of fibrosis therapies. In fact, we have way too many targets. And in fact, one of the big challenges is to select the good targets and to convince industry, and the interest is clearly there, that this is a good target and this is where we need the clinical trials. Again, the problem being that we don't have good endpoints, we don't have imaging modalities. Lung fibrosis is only developing in terms of clinical trials because you can have surrogate endpoints. You can have a CT scan. You can quantify fibrosis. We don't have that. We still have creatinine and kidney function. This is our big problem. And the first to develop an imaging method to quantify fibrosis in the kidney will be a hero as far as I'm concerned. And clearly this will be interesting and important therapy because so many patients who we see have, let's say, a glomerular disease, they have IgA nephropathy, so I could tell them, look, why don't we antagonize PDGFB? But at the same time, they come to medical attention first when they already have some degree of kidney failure. So the ideal therapy will target both the glomerular process and the interstitial process. And we should, of course, do this before it's too late. There's no way of getting this man over the water again. So clearly we can't come too late. It's not like the liver that has a high regenerative potential. So I hope I have convinced you that there are several processes where we can have new therapies. One of the most exciting areas being this crosstalk, and the mediators are starting to be identified, and maybe we will have a completely new approach to glomerulosclerosis there. We know proteinuria, and we know now many of the pathways how proteinuria induces tubular interstitial damage, and hopefully in the end we can develop new therapies to renal scarring. Thank you for listening.
Thank you very much, Professor Floege, for this excellent lecture. If there are some questions, we will allow them because we have some time. Please.
Thank you very much for a wonderful global information on the malfunction of the glomerulus and the kidney. I have two questions. I fully agree that you do have committed parietal cells which on specific demand replace the damaged or normally dying out podocyte. Where do these committed parietal cells come from? They should come from a stem cell population. Is this through the blood flow? Is this through the juxtaglomerular apparatus? Yeah. Where could this possibly be? This is one question.
You're right. This is the €1 million question. I think in ontogenesis, the parietal cells that, I mean, clearly parietal cells and podocytes have a common origin. For some reason, half of the cells in ontogenesis seem to be committed irreversibly to become podocytes, and this is like a limited pool. So where the other podocytes come from, I honestly don't know. I think the evidence, there was one study, Raghu Kalluri's study, describing that podocytes are regenerated from the bone marrow. I don't think anybody can reproduce that, and I think this is very unlikely, even though Raghu now has a clinical trial with bone marrow transplantation in Alport syndrome. But I would not like my kid to be in that trial. So essentially, we don't know.
And the second question is, you mentioned about damage by parietal cell activation, which take over the podocytes, and they can create glomerulosclerosis or focal glomerulosclerosis. In this case, how do the committed parietal cells, which are the benign cells replaced in podocytes, separate themselves from the activated parietal cells? Why doesn't one population counterbalance the damage a bit?
I'm sorry, the acoustics is bad. I'm not certain I got your question right.
How do the committed parietal cells separate from the activated cells which take over when damage occurs in the glomerulus?
I'm not sure that there's committed parietal cells. There's committed podocytes. Yeah. The parietal cells are still a relatively flexible cell population and of course, much more proliferative than podocytes. And this may be one of their, let's say, key advantages in taking over the glomerular tuft.
Thank you very much.
So the big question is, what keeps a normal parietal cell quiet? And what prevents them from doing this all the time?
Okay, thanks.
Is there Womenos Zakiris? Professor Womenos?
Thank you very much, Professor Floege, for this excellent lecture. I want to ask you, you showed us the interrelationship between podocytes, parietal cells, mesangial cells, and all the cells in the glomeruli. On the other hand, we know that tuberointerstitial injury is the main point. Is there any relationship between podocytes and tubular cells, apart from proteinuria? Is there any evidence that
Yes,
any one of chemokines?
That's another very interesting issue. Indeed, there probably is. One of the observations that we made is when we identified this parietal cell-specific promoter, some tubular cells also have it. So there's a cell population in the tubule that seems to have a similar program like the parietal cell. And indeed, we just showed that following acute kidney injury, it is this particular cell phenotype which gives rise to a regenerating tubular system. So and to answer your question, the big theory now is that there's a common program that is induced in the activated parietal cell and induced in the activated tubular cell, and maybe if we interfere with this common program, we have a completely new approach to kidney disease.
Thank you.
Dr. Zakiris, your question? Thank you.
Thank you very much for a very convincing and a very clear presentation.
Thank you.
I have said that before. It is pathophysiology as you have delivered it is so convincing and is so clear. My question, I think I have asked you that before. Why in clinical practice we don't see any drastic intervention since pathophysiology seems to be so clear?
Well, the pathophysiology is clear in some parts, but as I said, I talk to industry a lot, and one of my fellows is now the head of kidney research in Bayer, so he thinks along the same line. First of all, nephrology is not really interesting for industry. They want oncology. They want diabetes. Second, we don't have good endpoints. We don't have 30-day mortality. We don't have half a year of survival. We have difficult endpoints. Third, we have no surrogate markers. Cardiologists have CK, CKMB, troponins. We have creatinine, 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 creatinine. So we need better markers. We now have discussions with the FDA to accept at least proteinuria as an endpoint in membranous nephritis. This would make our lives 10 times easier in organizing clinical trials. So I think on the clinical side, we still have fundamental problems. The interest of industry is there, and it's increasing. And the good situation is that many blockbuster drugs from the cardiology are now expired in terms of patents, and industry is looking for new markets, and kidney fibrosis seems to be something interesting, so let's look at it. But then they talk to us and we tell them, well, you need a few years and you have creatinine clearance, and their interest disappears. So we need better clinical markers.
Thank you. Is there another question? Professor Floege, we are fascinated with very interesting results. Thank you very much.